makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas. Ating abutin ang pangarap ni Juan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas.
Ang pangarap ko ay maging piloto.
Pangarap kong magkaroon ng chief alternative for personal hygiene.
Pangarap kong magkaroon ng mabilis at murang transportasyon para sa lahat.
Pangarap kong masagot ang malnutrition.
Pangarap ko pong magkaroon ng effective communication means for emergency.
Pangarap kong ma-maximize yung renewable energy source and to reduce the carbon dioxide emission.
Pangarap ko pong maging scientist.
Ngayon na o simulan na, humanda sabay-sabay akyat. Hawak kamay tayo'y angat, lipad. Lipad. Ating abutin ang pangarap ni Juan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas. Ating abutin ang pangarap ni Juan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas. Maayos na bukas para sa Pilipinas. Maayos na bukas ating iparanas sa bayang Pilipinas.
Hello there! Magandang araw at magandang agham sa ating lahat. Welcome to the 20th installment of our Virology and Vaccine Institute of the Philippines, or VIP program webinar series. I am Landley Susi from the Environment and Biotechnology Division of DOST-ITDI, and I will be your host for today. For today's lecture, we shall be learning about immunoassays, which is practically an extension or a concrete example of a diagnostic tool used in virology. This webinar is part of the Virology and Vaccine Institute of the Philippines, or the VIP's information and education campaign on virology and other related topics or matters. This learning session is made possible by the collaborative efforts of the following agencies: DOST Central Office, DOST-ITDI, DOST-PCHRD, DOST-PICAORD, and the DOST Balik-Scientist program. I am sure that everyone here is excited to learn today. Am I right? So let's first check the number of participants in our Zoom meeting and as well as with our YouTube and Facebook live streams. So far, we have currently 77 viewers or participants in our Zoom meeting and 21 viewers in our FB. Thank you everyone for coming and joining us here. Before anything else, just a reminder for our Zoom participants, kindly rename your name or rename the format of your name to the prescribed format, which is your affiliation _ your full name. For example, I am affiliated with DOST-ITDI and my name is Landy Susi. So it will be DOST-ITDI _ Landly Susi. And also, For the information of all our participants from Zoom, Facebook, and YouTube, especially to the new ones here, you will get a copy or you will secure a copy of your certificate as long as you answer the evaluation form, which will be given at the end of the session later. Lastly, we would greatly appreciate if you will be conscious on muting your microphones for the whole duration of the program as a show of respect to our speaker and to avoid any interference to the audio. As a kickstart and to know more about our Virology Institute and the special role of our Balik-Scientists, let us all watch the briefer videos for the VIP and BSP program.
Recognizing the critical role of science and technology in economic development and progress, the Balik-Scientist Act or Republic Act 11035 was signed into law last June 2018 by President Rodrigo Roa Duterte.
This is actually putting into law a program that has been started by the Department of Science and Technology almost 40 years ago. But we need some legal support so that we can implement it in a better way. The Balik-Scientists will have an easier time in terms of coming here to the Philippines and rendering services.
Who is the Balik-Scientist?
Experience and expertise of Filipinos who have made good practice of being scientists abroad, so that they can share whatever they have in terms of knowledge and wisdom to our own institutions, our own researchers.
My role and responsibility of a Balik-Scientist is to be not just a teacher or a facilitator, but also a pusher of innovations within the program.
Balik-Scientists are given support by the government for their stay in the country and are likewise provided with a wide array of benefits to ensure their maximum output.
The best incentives or privileges or benefits are having to be exposed with our farmers.
My greatest privilege would be doing collaborative work with fellow Filipinos.
The DOST, as the leading agency in charge of the Balik-Scientist program, is tasked to facilitate the placement of the Balik-Scientist among its priority areas from its sub-agencies. P-Card, PCHRD, P-Shared, Partnering with the DOST are the host institutions, private or public entities, providing the appropriate resources to the Balik-Scientist in the completion of their research activities and other tasks.
I think the role of the institution is to give the space or the laboratory needed for the program or the project.
Working together, the DOST, the Balik-Scientists, and the host institutions have proven the importance of collaboration and cooperation, critical of any nation's vision for success.
I am an advocate of Balik-Scientist program. Okay, being one, I really truly felt that Balik-Scientists would be able to help in uplifting the economic growth of the country.
My hopes is to really contribute to the space agency. It's very, very pragmatic, and we will need everybody's help, and also promulgate the STEM program here in the Philippines.
We really need to encourage other Balik-Scientists or other scientists abroad to give their time. They need to give back and help the country.
With the enactment of the Balik-Scientist Act, the country is looking towards a stronger and more solid science and technology foundation, propelling the nation to further heights. Change has come indeed for science and technology. Science for change. Science for the people.
We are now living in a new normal. This COVID-19 pandemic marks as one of the global challenges experienced in this generation. It forces every sector of our society to innovate in order to move forward. We, at the Industrial Technology Development Institute of the Department of Science and Technology, is trying every possible ways to continue our service to our people, without compromising the safety of each and everyone.
All right, that was our video about the VIP program and also the briefer for the DOST Balik-Scientist program. I hope you get an overview or the gist of what we are currently doing or the efforts of the Department of Science and Technology in the establishment and also the special role of our Balik-Scientist in the establishment of the Virology and Vaccine Institute of the Philippines. So before we formally begin with the webinar proper, let's first have a recap of the previous webinar last Monday, February 21, given by none other than the husband of our resource speaker today, Dr. Cesar Elpidio Nadala, who is also one of our Balik scientists from the United States of America, who provided us a lecture about diagnostic virology. Dr. Cesar Nadala last Monday gave us a briefer of the full stages of diagnosing a viral infection of diseases putatively presenting as a viral infection. He started by describing virus as an obligate intracellular parasites with either a DNA or RNA genetic material enclosed in potent coat and at some cases with a membrane which is mainly composed of lipid. And also, he also mentioned that viruses come in a variety of shapes and sizes. Then, he proceeded to tackle the symptomatology or clinical manifestations of viral infections as a primary basis for physicians to begin diagnosing a suspected viral case or viral infection. He also mentioned that the majority of the immunopathologic signs are dependent on the host response to the presence of the virus within the system, which can be broadly attributed to the B cell and T cell mediated actions of the body. Succeeding, he provided an overview of the full spectrum of the different methods used in diagnostic virology, such as cell culture, which relies on the growth of viruses and suitable cell lines, laboratory animals or embryos such as that of chicken egg. Second, on the physical examination of virus particles through morphological characterization as examined through by observing through electron microscopy. Third, about serology, which is about the presence of antibodies and viral proteins present in serum or body fluids. Examples of serologic techniques that was discussed are lateral flow assay or LFD and ELISA, which relies on immunochromatography and precipitation, and also with the detection of viral antigens such that of SARS-CoV-2 antigen test kit. Another is detection of viral RNA or DNA or nucleic acid detection. And lastly, the use of whole or new generation sequencing platforms in diagnosing viral or in ascertaining that a certain disease is caused by a virus etiologic agent. For today's webinar, we shall be learning a lot about immunoassays. But before we proceed to the meat of the session for today, let me formally introduce our distinguished Balik scientist and resource speaker for this morning. Our Balik scientist or resource speaker for this morning is none other than Dr. Lourdes Nadala. Dr. Lourdes Nadala is a microbiologist or virologist by training with over 15 years of experience in microbial systematics and culture collection related activities, and over 20 years of shrimp and human diagnostics, including ISO accreditation and successful regulatory submission of in vitro diagnostic medical devices. She spearheaded the diagnostic for the real world's efforts for ISO 13485 accreditation and regulatory approval of in vitro diagnostic or IVD products, which include molecular point-of-care tests for early diagnosis of HIV in infants, viral load monitoring, and recently, SARS-CoV-2 tests using the SAMBA platform. She also has extensive experience in field evaluations and clinical trials involving in vitro diagnostics or IVDs. She also worked on detection methods for infectious pathogens, and spoilage mechanisms affecting food safety, ISO accreditation for 9001, and ISO Guide 34, reference materials, AOAC validations, and customer-driven research projects related to food safety and shelf life. So now, let's give a warm welcome and a big virtual round of applause to our resource speaker, Dr. Lourdes Nadala. And I will be now handing over the stage to our speaker, Dr. Lou. The virtual floor is now yours. Dr. Nadala, you're muted.
I'm sorry. Thank you very much, Lindley, for that warm introduction. And good morning to all our participants for this morning's webinar, which is part of the VIP webinar series. And as Lindley has mentioned, last week's seminar is sort of touched some of the aspects of immunoassay for virus detection. So today I will give more details about the immunoassays in the context of virus detection and diagnosis. As you all probably know, immunoassays are not only limited to virus detection, they're being widely used in the pharma industry and in drug detection, for example, in the field of biochemistry. So immunoassays are widely used in or across different industries that involve antigen-antibody interaction. So for today's seminar, I would give you a brief background on immunoassays and the principles of immunoassays and the major reactants, or what are the major components of an immunoassay, and also a brief background on monoclonal antibody production, as well as polyclonal antibody production, simply because these antibodies dictate a lot of the qualities or for example, the sensitivity and specificity of your immunoassay. So I'd like to give you a sort of like a background on monoclonal antibody and polyclonal antibody production. And also I'll give some types of immunoassays and examples that are being used in virology or in virus detection. This slide may be familiar to you. This slide shows you the series of events that happens during a viral infection. So as soon as the virus enters the cell or the body, this initial curve here represents the increase in the virus titer from the onset of symptoms. And then, let me just remove this part here. So at the onset of symptoms, or even before the onset of symptoms, the viral RNA is, there's a rapid production of viral RNA, and then followed by a production of antigen. So the viral RNA becomes a template for protein synthesis of the viral components. And so you have an increase in the viral antigen production. And after the onset of symptoms, like maybe 14 days, you would have, or maybe between 7 and 14 days, you will have the initiation of antibody production. So you have the IgG production and then the IgM production. And so these antibodies are the ones that are being targeted mostly by immunoassays for virus detection. So from this figure, you would see right away that immunoassays could not detect early infection because the targets are usually produced at the later stage of infection. So the antibodies are produced between 7 to 14 days after the onset of symptoms. So in comparison to the nucleic acid or molecular test, which can detect the viral antigen or the viral RNA or viral DNA early on, even before the onset of symptoms, you might say that immunoassays are not sensitive enough to detect viral infection, which is true. But as you would, as I would show you later, it has some advantages that are also not being made available by molecular methods or molecular techniques such as RT-PCR or other detection methods for viral RNA or DNA. So what is an immunoassay? This may be too elementary for some of our participants or some in the audience, but for the sake of the students in the group or in the audience, I wanted to start with this defining an immunoassay. So immunoassay is a bioanalytical method in which the quantitation of the analyte depends on the reaction of an antigen, which could be your analyte, and antibody. So an immunoassay can be targeting either the antigen, which is specific to a virus, for example, or it can also be an indirect method to detect viral infection by measuring the amount of antibodies in your body, for example, in response to a viral infection or, for example, vaccination. So as an example for immunoassays, you're probably familiar with the antigen test for detecting SARS-CoV, or if you want to see whether your body has produced antibodies against the SARS-CoV vaccine, you can also check the antibody level, although it could be a qualitative test using an lateral flow, or you can do a quantitative method using the ELISA technique, for example. And because of the specificity of the antigen-antibody reaction, immunoassays are inherently specific. They're very, very specific. And then you can have high throughput. For example, ELISA plates can accommodate 96 samples in one run. And it has a high sensitivity for the analysis of wide range of analytes in biological samples. As long as you have antibody or antigen to a specific target, you can practically use these two major reagents to develop an immunoassay. So it's a very versatile tool for diagnosis or for detection of biological targets or chemical targets or anything that you can raise an antibody against. And so the major reactants or players in an immunoassay include the antibodies and the so-called signal generating labels and separating matrices. So examples of these signal generating labels could be an enzyme, for example. The antibody or the antigen is conjugated to for example, an enzyme or a radioisotope or a fluorophore, etc., depending on what your detection method would be. And separating matrices, for example, you have the lateral flows, for example, and then you could also have other solid matrices to be used as a sort of like a platform or a capture for your target when you develop your immunoassays. So just to refresh you on the antigen-antibody interaction, let me start by showing you a picture of an antibody. So an antibody has this heavy chain region, or it's called the FC. And then you have the variable region where your antigens would bind on this part here. And this portion here, which is called the light chain, has two components. One is the variable region, and the other one is called the light chain. So this site here is very specific to a single epitope or single component, for example, of a virus. So in the presence of the antigen that was used to develop the antibody, this specific antigen binds only to this monoclonal antibody, because it has a monoclonal antibody has a single epitope or recognizes a single epitope. So an IgG has two of these binding sites here, but other isotype such as the IgM, which has 5 arms, could have 10 binding sites for a specific antigen. So just to repeat just what I said is, for a monoclonal antibody, it only binds to a single type of epitope. So only this, which is represented by this triangle here. But the polyclonal antibody means that it has several clones or different types of antibodies which would recognize different epitopes or different antigenic sites on the target. So in a way, polyclonal antibodies have more specificities, if you may, and so it can bind, for example, to a whole virus. If you have a polyclonal antibody, it's possible that different parts of the virus will be able to bind to a polyclonal antibody preparation, whereas a monoclonal antibody will only bind to one part of the virus. So this slide shows you, this is a very brief diagram of monoclonal antibody production. So you choose the antigen that you want to use as an immunogen, and you have to do a lot of research when you choose the antigen. And choosing the antigen involves a lot of consideration. So for example, do you want to use just a portion of a certain protein, for example, on the virus? Or do you want something that's widely distributed on the virus surface, for example, and so forth and so on. And once you have chosen your antigen, you prepare a purified antigen, because if your preparation is not pure, your mouse will produce different kinds of antibodies in reaction to whatever you inject or immunize it with. So to be able to have some sort of like very specific population, you would try to use a purified preparation as much as possible. And you can do boosting in between before you decide to sacrifice your mouse. But the boosting again would require careful research, whether you would like to, for example, boost with an alternate type of alternate preparation of the virus. For example, if you use the whole virus in the beginning, would you like to just boost it with just a specific portion of the virus? So you are trying to enrich the population that you would like to the mouse to produce. And so there are a lot of things that you can consider. And so this is just to cut the long story short. At the end of the immunization period, this can be determined by determining the titer in between. If your antibody is already in very high titer, you would like to harvest the spleen of the mouse and then do fusion with myeloma cells. So your antibodies are finite. So you want to have, for monoclonal antibodies, which is one of the advantages of mAbs, is it has an infinite life. And this is because the spleen cells, which were harvested from the mouse, which produces your antibodies, are fused with an infinite cell line called the myeloma cells. And once you have your fusion completed using with for example, with the aid of a polyethylene glycol, then you would have the so-called hybrids, right? So the hybrids would be the spleen cells which have fused with your myeloma cells, and the population of the hybrids can still be a different clones or polyclonal. So what you do is you seed these fused cells into a selection medium, which they call as hat, and then you isolate the clones. Usually, you grow this in micro titer wells, so you can process different. A number of hybrids as much as you can, because you don't know. It's like trying to find a needle in a haystack. So you need to grow your hybrids, and already at this stage, you also want to see whether these hybrids are robust, or they're able to grow compared to their other sister clones. And then when you do this, it's always advisable to freeze down cells even at this stage, because you never know what happens downstream. So it's very important to preserve your hybrids in liquid nitrogen, preferably, and proceed to cloning. So when you have sort of like chosen the clones that are, or the hybrids that are growing very well, you can try to determine whether the hybrid itself is producing the antibody that you prefer or that you desire. So at this stage, you already do something which is already considered an immunoassay by trying to detect for the presence of the antibodies that you prefer. So for example, if you're trying to look for an antibody against the spike protein of SARS-CoV, for example, then you will try to screen the hybrids using an antigen-coated plate. The antigen is coming from the envelope or the surface protein from the envelope of SARS-CoV. And once you detect the presence of a reaction, for example, by ELISA, then you can choose those clones or hybrids, and then try to purify specific clones using the limiting dilution technique. So what you do is you do several dilutions of your cell suspension, and then plate them on a 96 well plate. And because you have diluted your samples to a certain, like very low cell count, then you will be able to isolate single clones when you do the subcloning process. And once you have your subclones, again, you will try to screen them, whether they are producing the antibody that you are targeting for. And once you have chosen your the best clone, then you can proceed and produce your monoclonal antibody. But just to give you some tips, it's always good to, again, to freeze down different clones at this stage when you have the individual clones that are producing the antibodies that you desire, because as you go downstream, some of the clones may not be robust enough and grow well during the transfer or subcloning. And so you don't want to repeat the whole process of, immunizing again or all this very tedious and very long process. So it's always good to preserve, to freeze down cells with at the hybrid level and then at the clonal level. So when you have your individual clones that are already producing a single population. And so the hybrid or the clone that you have chosen as the best, for example, you can either proceed with in vitro cultivation, which is like a semi-continuous process, or you can also inject the whole the hybridoma or the monoclonal antibody clone again into a mouse and collect the ascites or the acetic fluid. I think this one is now being really discouraged because you don't want to sacrifice too many mice, right? Although this is really a very productive way of producing your antibodies, because you will produce tons of or milligrams of antibodies when you inject it directly to a mouse. However, as I said, you don't want to unnecessarily sacrifice mice. You have the option of growing it in cell culture tanks or in what you call a cell line flask, for example. And this, you do a regular harvesting and feeding of your cells. So when you have your clones, you do a lot of studies, not just a immunoreactivity, but also looking at the cells, whether they are floating cells or they are adherent cells, because that would also give you an idea when you try to produce your monoclonal antibodies, for example, in this bioreactor. And so this is what we currently use in our company because we only need a small amount of antibody. So this bioreactor can actually be used, like if you're very good in your aseptic techniques, you can maintain this and use this for like 6 to 7 months. So what you do is at the layer of this flask is a compartment where you inject your clone, and it has its own medium, and it is porous in the sense that the medium outside, which is actually also nourishing the cells, are able to, the nutrients are able to go through the cell compartment. And so this nutritional medium here, we change it like twice a week or 1 to 2 times a week. And because it has an acid indicator, if your cells are growing really well, this turns into orange. You don't want it to grow into overgrow, changing the medium into yellow. Or if your cells are not growing very well, this can be a pink medium, which means that there's something wrong with your cells in the cell compartment. So once you have that's for the monoclonal antibody. For polyclonal antibody production, usually this involves animals. So you don't want to use mice, because then when you finish or at the end of your immunization, you will only be able to harvest so much serum or blood from the mouse. So what we have used in my previous company was raising polyclonal antibodies, for example, in goats, because goats can give you hundreds of milliliters of blood where you can collect your serum and eventually the blood and eventually your serum for polyclonal antibody purification. So the steps are almost similar, except that you don't do any fusion of your antibody in between with a myeloma cell line. So this means that at the end of the immunization, when you have sacrificed your animal, your polyclonal antibody will have a finite, you will only have a finite supply. So once it's done, then you have to repeat the whole process again, and you may not be able to get the same high affinity antibody as the initial one, because antibody production is affected by the animal itself, the health of the animal, and then the antigen that you use to immunize. So once you have your serum from animals for polyclonal antibody production, or you have your monoclonal antibody supernatant, you have to do affinity purification to further remove the impurities from your preparation. So, as I have mentioned earlier, the quality of your immunoassay would depend a lot on your antibody, for example, or from your monoclonal or your polyclonal antibody preparation. So, most, if not all, who are developing immunoassays would go through what we call as affinity purification. And basically, you use a column which is made of either agarose or any other inert material, where you would, for polyclonal antibody production, where there are more impurities, you would like to conjugate your column to an antigen specific. So whatever antibody goes through your column will bind to the antigen in the column. So as I have mentioned earlier, that the reaction between antigen and the antibody is very, very specific. So all the antibodies that were generated against a specific antigen will be bound in the column as you pass through your serum through the column. So then, so this is the step where a mixture of different things goes through the column, which has been conjugated to a specific antigen to capture your antibody. And so once you have run through all your material, or your serum or supernatant in the case of monoclonal antibody culture, then you wash your column several times to remove the impurities. Washing meaning with a certain buffer. And then once you have completed the washing, then you would like to elute the antibodies from the antigen that bound these antibodies in your column. So it is a process that does not really affect the functionality of your antibodies. So once you do the elution, then you will have your specific antibodies. And then the column is left again just with the antigen that you had conjugated the column with earlier. And so this column can be used again by regenerating the the column itself and storing it in something that would prevent the growth of microorganisms. And for example, if you have a monoclonal IgG, there are columns that are already available in the market, and these are protein G columns, which are very good in separating or in concentrating your IgG. And depending on the volume of supernatant that you have, you can use a single column, or you can do a series of columns where you can pass more volume of your supernatant. So this is a very simple way to purify your monoclonal IgG. And as I have mentioned earlier, this can also be reused. So we try to reuse the columns because they're quite expensive. And there is a way to preserve the column and to also monitor whether it's still good enough to purify your monoclonal antibody or polyclonal antibody. So it is important to not lose your antibodies, because if your column is no longer working, your antibodies will just go through the column without being bound to the column, the column itself. So then you will lose a lot of your antibodies. So in our lab, we monitor the eluate after you pass through the column, and also at the end when you clean the column, we also try to measure whatever is eluted after you have collected your antibody. So there is a way to monitor the quality of your column, or whether you need to continue using it, or you need to replace it with a new column. So for immunoassays, sometimes you would hear the word homogeneous immunoassay or heterogeneous immunoassay. So homogeneous immunoassay, all the components are mixed in a single vessel or container where all the reactions take place. So an example is a lateral flow dipstick, where when you put everything in a reaction well, you don't do any washing or any separation. You just allow your sample to wick up the dipstick or the test strip, and then see whether there is a reaction in the test line and also in the control line. When you hear the word heterogeneous, it means that there is a separation. There are separation steps to remove unreacted excess of, for example, the analyte or the antibody, so that you remove the so-called background or noise or non-specific reactants. And an example is for ELISA. When you're running an ELISA, there's a lot of washing and rinsing steps to remove background materials or materials that may cause non-specific reactions. And there are also several kinds of let me see, how do I remove this? ELISA or enzyme-linked immunoassay, or any immunoassay. So you have the competitive immunoassay. So it is best described by this cartoon here. So you coat your antibody on a solid support, and usually it's a microtiter plate, the most common one. And then you add your analyte and labeled analyte. So the analyte is, for example, it comes from your sample, a patient sample. And then the labeled analyte is already a purified antigen, which is, for example, the S protein of the SARS-CoV. And it has been labeled with a, if it's an immunoassay, with an enzyme. And the labeled S protein, and then the S protein from the, for example, if there's a virus in your sample, then they would compete for the antibody binding sites, right? And so after you have added this, all the reactants, you do washing, as I have said earlier. And then once you have completed all the washing steps, the last bit would be to add the substrate for whatever label you used. So for example, if you used HRP, then or horseradish peroxidase label here, then you can add a TMB or tetramethylene methylbenzidine. And so you will see a colored reaction. And for TMB, you need to stop the reaction, otherwise it will just go on, and then at the end of your assay, everything will be yellow. So you need to add a stop solution, which is usually HCl. And then this is read in an ELISA reader plate, depending on the wavelength depends on the label that you used. And so because there is a competition between the patient's analyte or the patient's virus, if it's present, between that and then the labeled analyte. So if you have more of the labeled analyte, which is your reagent, then that means that the sample from the patient does not contain that many virus. So therefore, the signal is reciprocal to the concentration. So the more virus you have in your sample, then the signal becomes lower, because then all the labeled antigens will be displaced and will be washed off. Okay? You can have also, in the earlier slide, you use the antibody to coat your solid support or your ELISA plate. You can also have the reverse. Like, for example, if you're looking for antibodies, then you would line or you would coat your wells with the antigen. So the principle is the same, but this time you're looking for antibodies against the virus. So it goes through the same series of steps. And then again, if you have more antibodies present in your body, for example, then you would have a lower signal of the substrate or the reaction. So it's the same principle. It's just that you are using antigen to capture the antibodies present in your sample. So for this type here, you will be using blood sample for SARS-CoV, for example, or any other viral target, because you're looking for antibodies in that individual. So you use a blood sample. In the earlier example, where you are looking for the antigens, you have to use the sample that has been reported to have the most virus found in your body. For example, for SARS-CoV, then you use the swab from your nose and throat. And for HIV, because it's also bloodborne, you use also the blood sample. But HIV is known to produce a lot of antibodies. So usually the methods or the immunoassays that are used to detect HIV are looking for antibodies rather than antigens. Then you have the non-competitive immunoassay. This is pretty straightforward. So you have your antibody coated on your plate, on your well, or on a dipstick, and then you add your antigen source, for example, and the antigen just binds directly to the antibody present on your titer, on your well. And you do, again, a series of washing, and then you come back with a second antibody, which is also recognizing the antigen, but this time this is labeled. So this one looks like a sandwich, where your antigen is being sandwiched by one antibody here, which has a different specificity to this antibody here. So these are two monoclonal antibodies that you use. And in this case, the signal is proportionate to the concentration of your virus. So I sort of like simplified these immunoassays, but for enzyme-linked immunoassays or even other types, there is always this risk of having non-specific reactants coming from the sample itself, because the sample is really like for example, blood, or from your nasal swabs. It could have a lot of non-specific impurities, for example. So there's a lot of washing for ELISA plates, for example. And then you also need to do blocking using bovine serum albumin or milk, or skim milk. Skim milk is the most commonly used blocking medium. So you are able to coat the empty spaces here, so that other impurities could not bind on these empty spaces. So you want to block off all empty spaces by using a blocking agent. So what are some examples of immunoassays used for virus detection? So I have sort of talked a lot about enzyme-linked immunoassay or enzyme immunolinked assay, or ELISA, where the label is an enzyme, and the most common enzymes used are either horseradish peroxidase and alkaline phosphatase, or AP. Then you also have the immunofluorescent assay, where you have a fluorophore or a label that is that fluoresces at a certain wavelength. Then you have the chemiluminescence assay, which is almost similar to enzyme-linked assay, except that the labels use either the label is either emitting light itself, or the HRP. You can use enzymes also as label, but the substrate being used is different, such that once it's oxidized, it emits light that is read by the machine. Then you also have the immunoblotting assays. And as I would show you later, there are two types. One is quite labor intensive, but they have already sort of simplified this so that it can be used by without very intensive training. Because if you just use if you go through the usual immunoblotting like the Western blot, it needs a lot of training. But for example, for HIV, there's already a lot of Western blot strips that are readily available, that you only need to add your sample in and do some washing steps. But it's a lot simpler than the typical Western blot procedure that we have probably learned from school. And then you also have the Octor-Loney technique. I just wanted to include this because it's not widely used in virology, but it's still being used in some fungal diseases. But it's a very simple thing to do if you have an unknown. For example, if you want to look at the specificity of your antibody against two related strains or two related antigens, for example, it's a very simple thing to do that can be done in labs without, sophisticated equipment. And then the radio immunoassay, which is already not widely used because, as I would show you later, handling isotopes can be very challenging, and most especially the waste that you generate from doing radio immunoassay. So this is just a simplified version, a simplified diagram of developing an immunoassay. So you can, as I have already gone through earlier, you can have you can generate your monoclonal antibody in the lab, of course, with the help of mice. And polyclonal antibodies can be raised in goats, in chickens, and in mice also. So depending on what your target is and what isotype do you aim for. For example, for our IgA, they're best produced in chickens. And so we harvest the eggs from here, from the laying chicken. Then you can, once you have your monoclonal antibody, which again, which I would like to reiterate as the key reagent for your, if not one of the two key reagents for developing your immunoassays. And when you have, if you're developing an ELISA method, you have to do a lot of screening and validation to make sure that your ELISA is specific enough to only pick up or only react to your specific target, because you would imagine if you have an ELISA that would detect, for example, a SARS-CoV-2 and also another human coronavirus. So that would be giving you a false positive result for SARS-CoV-2. So then the doctor would have a different way of clinical management if you get a false negative result for SARS-CoV-2. So what I'm trying to say is, before you have your assay used, it goes through, if it's for human diagnostics, then it goes through a rigorous validation, both in the lab and also clinical trials, just to make sure that your assay meets the requirements of the national government or the national regulatory body. And if it's for example, for food pathogens, then it's probably less stringent than for human diagnostics, but still you would have to, for example, if you want it to be accepted, then it has to be ISO certified or compared against an ISO certified method, or it has to be approved by AOAC. So developing an assay takes a lot of, requires a lot of testing just to make sure that your assay is specific and sensitive enough compared to what they call as the gold standard, or whatever is the state of the art at that time. So let's look at some of these examples. So I have already talked or touched on ELISA, which is the most common immunoassay for viral detection. So I have shown you earlier where you have a direct sandwich ELISA. So the antigen is sandwiched between one antibody, which is called the capture antibody on the solid phase, and another antibody, which is linked to an enzyme. And then once you add the substrate to this enzyme, it gives you a color reaction. So this one is the most widely used. And you have an indirect sandwich where you have the same as this, but this secondary antibody is actually not labeled. So there is a third antibody, which is the one being labeled here, and then upon the addition of the substrate gives you a colored reaction here. So this method is requires more steps, but there are some advantages for indirect sandwich ELISA. And then you also have a sandwich ELISA with a streptavidin biotin detection. So if you notice here in immunoassays, you do not amplify the target itself. So there's a one-to-one correspondence, right? But if you use a biotinylated antibody, for example, where you can have several biotins, you can have 12 biotins, for example. So then the signal itself is going to be amplified because the strep avidin can bind to or the strep-avidin HRP can bind to different molecules of biotin. So for example, if you have 10, then the signal now becomes like times 10, right? So there is an amplification of signal if you use this approach. So for the most commonly used label, as I have mentioned earlier, is alkaline phosphatase or horseradish peroxidase. And the substrate that is commonly used for AP is PNPP or paranitrophenyl phosphatase, and TMB, which is for HRP. So I have used this quite a bit. It's very, you have a ready to use, or you can prepare your own reagent. So it's very, very handy. And this is just the steps that I'd like to show you. So with indirect ELISA, for example, where you use a antigen-coated well, there's a lot of washing in between. As I have mentioned earlier, this is to remove all the excess unbound molecules and also impurities from your antigen preparation or from your samples. So you have a cleaner background, apart from adding a blocking reagent also in your assay to further reduce the background or the noise. And in this case, the antibody is the one that's coated on the well, and it goes through the same steps of washing and before adding your substrate. So what are the advantages of ELISA? So it's quite sensitive and specific. Again, this would hinge on your the quality of your reagents, the quality of your antibody, or if it's an antigen assay, or if it's looking for antibody, the quality of the antigens that you have prepared. So if you have a lousy antibody, then you would also have a lousy ELISA, no matter how much you optimize. So it's very important to have a high affinity antibody, high avidity antibody, or you have to have a highly purified antigen if you're using it to capture your target. It has a very high throughput. As I have said, you can process like at least 90 samples, because you have to also run controls. So you can run 90 samples at the time, for example. And so it's also a well-established method. So it's almost like, you know, you don't have to do a lot of, if you're using, for example, a commercial immunoassay, you don't have to do a very intensive validation, because it has already been established, or it has been validated because it's an established method. And it does not require expensive equipment. You can do manual washing, but of course, you know, that can become labor intensive, right? But if, for example, your washer broke down, then you can do manual washing and still go ahead with your ELISA assay. And as I have said, it's a multi-step. There's a lot of blocking, washing, and all that. But this can be, if you do it manually, but you can, it's now, there's a lot of automated ELISA washers that are quite cheap. So then you can reduce the labor and time for these steps. And there are some that require overnight coating of your antigen, for example. So it may take longer than usual because you have to coat overnight and then you have to do the ELISA. So it would probably take like 1 to 2 days before you get the results. But there are some ELISAs also that are quite quick, but it's not really as quick as some molecular assays, because molecular assays, you can have results in 45 minutes to 1 hour, including sample preparation, etc. So these are some of the advantages and limitations of ELISA. So there's another immunoassay which is being used still quite a bit in virology, and this is called the immunofluorescence assay, or IFA. And the labels used are for your antibody are fluorescein, isothiocyanate, or you may hear Lexaflor. And these labels are fluorochromes, which fluoresce at a certain wavelength. And this requires quite some training, and also a fluorescence microscope for slides if you want to look at infected cells, for example, on a slide preparation. Then you need to have a fluorescence microscope. And if you are trying to do some cell sorting, then you would need a fax machine or a fluorescent antibody, a cell sorter. So which is quite expensive. So these are just examples of a dengue virus in viro cells, and you would notice that these two are FITC labeled, but you would see that they have different colors, and it's because the background staining was different for these two. And these are the virus the ones that are fluorescing here as yellow-green, and then the pinkish, deep pink here are the ones that are showing where the virus have ended. And this one is a different label altogether. And if you have it in a homogeneous mixture, for example, then you can add your viro cells, and then you add your virus and your antibody, which is conjugated to an immunofluorescent. Sorry for the typo here. Immunofluorescent label. And then look at it under the microscope, and so you would see something like this. Or you can go do flow cytometry to separate the cells that have been labeled with, for example, Lexaflor. So this is not, again, this is not widely used, but it's still being used for virus detection in infected cells. And chemiluminescent immunoassay, the label being used is a luminescent molecule, or it can be an enzyme, but the substrate being used generates a certain light, which can be seen in the visible or near visible spectrum. And so you have a specialized equipment to be able to use chemiluminescent immunoassay. So this is just a cartoon where you have, they mostly use magnetic beads, which have been coated with a specific antigen, and you're looking for antibody in a sample. And you have the same, there's a tracer, which is an antibody labeled with the isoluminol, which emits light, and this is read by the instrument or the machine. And I have mentioned earlier that it can be also an immunoassay or enzyme-linked immunoassay, but instead of using the substrate earlier for alkaline phosphatase, in this case, it's a different substrate, which upon oxidation emits light. So this is all being run in an automated machine mostly, and the chemiluminescent or CLIA machine can be also expensive. And another immunoassay is known as immunoblotting, and there are two types. One is where you blot your sample directly onto the membrane, and then the target antigen is detected with a specific antibody, which is conjugated. Sometimes they use gold so that you can easily or visually see the presence of your target. So it's a very qualitative and subjective way of doing things, but we do this when you're just in the early stages of developing your immunoassay, for example, where you want to just put a sample where you can do spike samples and then just blot it onto a membrane, for example, nitrocellulose, and then use your antibody, labeled antibody, and with gold, so you can already see right away if it's really there. There is another immunoblotting technique or method, which is called the Western blot. And it's a two-step. So you have to run your sample in an SDS gel electrophoresis to separate your proteins. So at the end of this whole process here, you would have an agarose gel, SDS polyacrylamide gel, which would contain all the protein bands. By the way, this is just for the purpose of showing that there are supposed to be bands here, but this is the marker where you have the each band represents a molecular weight marker. And this one is colored, I'm sorry. But your the wells here would represent, for example, different patient samples. And you wouldn't see anything here at the end of the SDS gel electrophoresis, except the markers, okay? Then once you have your gel, you would blot it into either a nitrocellulose again, or you can have a PDF membrane. And some people use an electro transfer into the membrane. But you can also do like a simple transfer. You do have to apply a current, and then at the end, all the proteins on your gel would have blotted or transferred into your membrane. And again, there would be a lot of washing in between here. And then you do also blocking, either with BSA or skim milk. We usually use skim milk because it's cheaper. And then incubate the membrane with your primary antibody specific to your target protein, for example. So if you already have your primary antibody, for example, it was produced in mouse, okay, then you can put your you can incubate your membrane in that primary antibody. Usually it's about like an hour, 30 minutes to an hour. And then the membrane is again treated with a secondary antibody, which is HRP labeled. And again, a lot of washing here. And then at the end, you can either do a chemiluminescent HRP substrate and expose the film, expose the membrane to a film. So you would have a like an X-ray kind of thing, or you can have you can just if you have used an HRP which is labeled to an or a substrate which is colored, then you will see the colors also of these target proteins if they are, or antigens if they're present in your sample. So this is a very long process. So it's not advisable to use it unless it's really a very key immunoassay that you want to really confirm. Because this one is very, very specific because you have separated the proteins and you are really seeing the target that's a very specific antigen or specific protein from your sample. So this is a very good test to confirm. In fact, Western blot is being used to confirm if you are positive for HIV antibodies using 2 rapid tests. WHO requires confirmation using a Western blot, and so it's a good procedure to have. But as I have mentioned earlier, there are already commercially available Western blot strips, for example, for HIV. So you don't have to do the running the gel and all that stuff, and then blotting the membrane onto the membrane and doing the detection. So what you do is you just get those strips and then incubate it with your serum, for example, and then do the washing and then blocking, etc. And then at the end, you will see if your patient really has HIV, then you would see the target proteins, which are in this case for HIV, Western blot, they look for P24 and GP36, and there are other marker proteins that they would look for. So it's easy and it can be done by untrained technicians, or doesn't require a highly skilled technician. And then Octoloni double immunodiffusion. This is a very simple thing to do. So as I said, it can be done in your lab. If you are looking at antibodies, for example, you have an antibody and you want to see whether it would has specificity. For example, you have 3 different SARS-CoV, for example. So you have SARS-CoV-2, SARS-CoV-1, or SARS-CoV-3, MERS, or other human coronaviruses. So what you do is you prepare an agar plate and then you bore some wells. So you have one in the middle, and then you can put about 6 here, or you can have micro wells. But essentially what you do is you put your antibody in the middle, and then you put your antigens on the side. So it's like a radial thing. And then let the reaction proceed, and at some point, if your, for example, if your antigen here is exactly the same as here, then you would see something like this. So it's it's like a precipitin line, which is very obvious to the eye. And if one of your antigen is different from the other, then you would have like, they call the spur here. So they are not exactly the same antigen, okay? And if these two are totally unrelated and are not, don't react to this antibody, then you would have something like this. So it's very easy to do and very fast reaction. But as I said, it's a very qualitative and it can be subjective too. So it's used mostly for screening, but not really for diagnosis. But except I think for some fungal diseases, which are still using this type of immunoassay. And then lastly, we have the radio immunoassay, and this was developed in earlier than 1977. I forgot the year, but they received a Nobel Prize for this one. It's extremely sensitive because it can be used to detect minute concentration of, especially for hormone levels in blood. So it is directly proportional to the amount of analyte present. So if you have, it's like the graph for the non-competitive assay, where you have like a sigmoid curve. And however, as I have mentioned earlier, radioactive materials are very hazardous, and require special arrangements for storage and high cost of waste disposal. So there are also some difficulties in the automation of this assay. So it's not being used as widely as before, because now we have more options which are less hazardous and easier to handle. Oops, sorry. So this is just a pictogram of radioimmunoassay. It's similar to the other immunoassays, except that the label is a radioisotope. In this case, it's iodine. And to read the radioactivity, you have to use a gamma counter. And for other labels, you have to use a scintillation counter. So for example, for radioisotope carbon or hydrogen, then you have to use a scintillation counter. So these are quite expensive, and also, as I said, they're not as widely used as before. So in conclusion, while immunoassays do not offer the same sensitivity as nucleic acid detection methods or molecular methods, particularly at early stages of infection, they come with the advantage of a reduced cost, reduced complexity, and higher utility for use by untrained personnel. For example, lateral flows can be run by anybody with very minimal training. And so in our work in Africa, the microscopists, the lay workers can run the lateral flow assays. And also because the analyte being detected is part of the virus or part of a whole virus, it can be an indicator for active infection in acute infections. But if it's like chronic infection, like hepatitis B antigen, hepatitis B virus, for example, or HIV, then it's not true, because you will always be shedding the virus, because it becomes integrated in your chromosome. So that means it's not really active infection, because for example, for HIV, you may have 1,000 copies of our DNA or RNA in your system. You may always produce a virus, a whole virus, but it is not an active infection. Thank you. And if you have any questions, I'd be happy to answer them.
All right. Thank you, Dr. Nadala. That was a very comprehensive and very informative presentation about immunoassays. I'm sure that our participants were enlightened and gained a lot from this lecture. Okay. I am sure as well that our participants are excited to ask their questions to our esteemed speaker. So if you have any questions in mind, kindly type it in the chat box. Over the Zoom platform, and for our FB, maybe for our FB, they could also type it as a comment, and the moderators will sweep through those questions and flash it on the screen. Okay, do we have a question now? If we currently do not have yet a question, I think I will be the one throwing the first question. So my question is, do you think that, I believe that the pregnancy test kit is also an example of an immunoassay. Aside from the usual immunoassays for infectious disease, do you think that the pregnancy test kit is also one of the most widely used immunoassay?
In a way, yes. As I have said, immunoassays, for example, lateral flow dipsticks or test strips, are being used not only for infectious diseases, but in a whole range of different, even metals or even other materials that are where you have antibodies prepared against, then you can have an immunoassay.
Another question: do you think that we could prefer to use only one type of diagnostic test, for example, immunoassay alone, or it depends on a case-to-case basis?
It depends on the target, and also on the pathogen that you're trying to test. For example, in HIV, in the initial diagnosis, where you have a positive result in one lateral flow, they would like to confirm with another lateral flow, and then confirm with Western blot, or for more developed countries, of course, you have the molecular technique or the molecular test. And this is because if you have a false negative result, then it would really impact the care that the person would get. And also, like, for example, stigma and all that stuff. So I would think it depends on what the disease is. And it also depends on if there is a follow-up for that particular disease. It's always good to compare with another method if you get a positive result. But if it were, like, for example, if it were, let me think of like chlamydia, or something that's not life-threatening, and although it has some bad effects if you don't get the diagnosis right. For chlamydia, for example, you can have infertility. Then I would think that having a second test, it's like having a second opinion for a test for a disease is always good to have.
Okay, I have another follow-up question. I think, could you give your perspectives on when would be the most good timing to use an immunoassay compared to a nucleic antigen test or the nucleic acid based test?
I think if you want to see or determine whether the infection is active, if it's an acute infection, okay, as I said, you should not do this for chronic infection because you will always detect the virus. So that means you may be misled that it's an active infection. But for acute infection, like for example, SARS-CoV, if you want to make sure that you still have active infection and highly infectious, then an antigen test would be very useful. So that's why now, coming to the States, for example, they do accept antigen testing for SARS-CoV. Unlike before, they would only accept RT-PCR or molecular test. Because if you have a positive antigen test, then that means you have really an active COVID infection, which could be very transmissible. Okay.
Okay, thank
unlike,
you.
Unlike molecular tests, it could be you could be positive for the nucleic acid, but actually it's just a inactivated virus, or it's no longer infectious, especially if it's like, towards the tail end of infection.
So another question, what could you say about, because even for veterinary disease experts, they actually label diagnostic tests as gold standard.
For veterinary?
Or even humans.
The gold standard depends actually on the test itself. So most commonly reported viral infections now, they consider the molecular test as the gold standard, but there are still some diseases which use immunoassays as the gold standard. And these are the more difficult viruses or viral infections to culture, for example. So as I said, immunoassays depend a lot on antibodies. So if you don't have the antibody, then molecular tests would be the diagnostics that would be considered also as gold standard. Or you cannot develop an immunoassay unless you have isolated the virus or grow the virus. So it's a or you can have like components of the virus synthesized as antigens, or parts of the virus can be synthesized as antigens, and you can use that to immunize and produce your antibodies.
So we have another question from Patty from the University of the Philippines Diliman Institute of Biology. Her question is, what type of immunoassay is the antigen test for COVID? Does the reliability of the antigen test decrease as more variants arise?
I think the antigen test, again, would, there are so many antigen tests in the market, if you notice, for COVID, right? But they all have different sensitivities and different specificities, simply because they have different antibodies, or they use different antibodies, right? So for variants, if the mutation is something that does not affect the amino acid that's part of the binding or of the virus that binds to the antibody, then even if it's a genetic variant, it may still be detected by antigen test. But if the variant itself, like for example, this the Omicron, where you have so many mutations, which could potentially affect the protein itself, then it may be affected in its ability to bind, or the antigen test may not be able to detect this virus. So even molecular tests, we as a manufacturer have to always do a blast every month for all the mutants out there in the database, to make sure that our primers, our detectors, our capture probes are still able to detect the variants. So I'm not sure if it's being done for antigen tests, but I hope they would also do that, because, or at least they give panels to make sure that the antigen tests continue to pick up the variants. So there's one question here about using horses for polyclonal antibody production. Horses are quite expensive, so I think the biggest animal that they use is the llama, or because horses are being bred and they're quite pricey, right? So you don't want to use a pricey animal to produce your antibodies. Goats are good. Then you can use the meat after, you can still use it for food consumption.
So
Then,
another question. So good morning from John Paul Ramos from the De La Salle Medical and Health Sciences Institute. He asks, allow me to read. Good morning. Thank you po, Dr. Nadala, for the very informative lecture. And his question is, which immunoassay or assays would be best for the evaluation of the responsiveness of immune cells against an antigen that is to be used as a vaccine?
I would suggest, I think they should use the ELISA, because if you are going to screen so many cells from the sample, then you would need to have something that's high throughput, and at the same time, you're able to use a very sensitive and specific test. But again, this is on the assumption that there's already an ELISA, that's a very sensitive ELISA for this purpose. Otherwise, if you have to develop your own, you have to make sure that you use a very good antigen when you're trying to, antibody when you're trying to screen antigen to be used for a vaccine, for example.
So we still have a lot of time. Maybe we could entertain 2 or 3 more questions. So for our participants who have something that they want to be cleared, or a question that they want to be cleared from our speaker, you could actually type in your question again in our chat box. So if we do not have any question from our participants, I think I will be throwing the last question.
Okay.
So I think, which would you think would be the best diagnostic test for monitoring or surveying an epidemic?
It would really it would really depend on target or on the purpose of the survey. So, for example, if you want to monitor a patient, for example, with this is in the context of a patient with HIV, right? You want to monitor the patients or the patients in a cohort to be able to see whether the therapy that you gave to these patients is effective against HIV. Then you want to use a molecular diagnostic assay for HIV monitoring, because you want to see the virus in the free virus in the plasma. Okay, because if you use something that's like antibody, you cannot. They will always have an antibody to HIV because they have chronic infection, right? Once you are HIV positive, you will always be HIV positive for antibodies and for the virus itself. The same thing for hepatitis B virus. So if you are monitoring, for example, the SARS-CoV-2 infection in a community, and the purpose is to really see if there are still infected individuals, then you may want to see. It depends on that community. If that community had COVID-19, very high prevalence of COVID-19, you don't want to use an antibody test, right? Which is an immunoassay. But you may want to see you may want to see an antigen test if you want to make sure that they're no longer infectious. But if you want to also see whether they are asymptomatic carriers, for example, then you would like to use a molecular test. So it depends on what the goal is for the monitoring of the disease in a particular population. So it depends on the population itself, the prevalence or the exposure of that population. So a lot of factors have to be considered when you want to decide which diagnostic tool you would use.
Great, thank you, Dr. Nadala. Actually, we have another question. I think this will serve as the last. It's again from Patty Tamesis from UPD Institute of Biology. How does an immunoassay produce a false, positive, or negative result?
Okay, so for example, in an ELISA assay, if you have a specimen that has, like, for example, high IgM content, like people in Africa, these isotype tend to stick to your 96-well plate. So it's very hard to remove them. And so because they stick very well, then your reactants would also be sticking onto this non-specific material, right? So if they're not washed off, then they would be giving a false positive reaction, because when you add the other components of the assay, because they are there and they have stuck to the well, for example, then they would be reacting to your antibody, because the antibody would stick non-specifically to that sticky material. And so when you add your substrate, then it gives a colored reaction. So it would look like it's a positive result, but in fact, it's a false negative or false positive result. False negative is if there are some interfering substances in your sample. There are known substances which interfere with the assay, so they would also block the antibodies or antigen, for example. So there would be no interaction between the antibody and antigen, and so you will not be able to detect that the analyte is there because there is an interference from other materials. So you may get a false negative result.
All right. Thank you very much, Dr. Nadala, and to our participants. What do we have? Should
one
we
more
entertain the
question.
last? Yeah. What are? Could we entertain? Could you flash back the other question? I think we could entertain that one. So, what are the chances and factors that result in immunoassay interference? From Jasser Ramirez of Carolina State University.
So when you develop your assay, we have this we call the interfering panels. So what could be interfering in your assay would be, for example, the medications that you take, or for example, when you were tested, you were taking antibiotics, which sort of like removed for example, if it's a bacterial infection from your body, or shedding was the word I was looking for, then you would have a false negative result. So in assay development, we do study all the potential interfering substances, like, for example, rheumatoid factors or antibodies from autoimmune diseases, which tend to be really very sticky. And so there's a whole lot of panels that you can look at to screen for potential interference with your assay. And that's always part of your validation, and whatever submission you have to make, you have to test them against these potential interfering substances. If, for example, you find some of them interfere in your test, it doesn't mean that you cannot market your test, but you have to put this under limitations of your assay, so that the tester or whoever is using the test would ask them, did you take antibiotics, or do you have an autoimmune disease? So these things are factored in. So you have to put it under limitations and warnings of your diagnostic tool.
We have another question from our audience. Do we have to entertain this one?
What are the main obstacles when optimizing your immunoassay in terms of sensitivity? There could be a lot of factors, but I think the first one is really your antibody, your antibody may have a low affinity to your target. So because that affects sensitivity, okay? That's the first one. It's the quality of your antibody. And then the second one is when you are optimizing your, for example, ELISA diagnostic device, you may want to look at different, for example, blocking agents. So you have to try different concentrations and different buffers, because it depends. Your ELISA would depend on a lot on pH also. So you have to consider all this, because it would depend on your sample. For example, if your sample tends to be acidic, then you may have to do a neutralization in the beginning, and so forth and so on. So it's a whole lot of things that you have to consider when you're optimizing your immunoassay. But the first or the key thing is your antibody should be really of good affinity and avidity.
So, and that's it. We are done with our question and answer. Thank you very much, Dr. Nadala, and to our participants who actively engage in our Q&A session. In the interest of time, we will be ending the Q&A session now. So if you have any remaining questions that has been unanswered, we will be sending them directly to Dr. Nadales via email. So let's proceed with the awarding of the Certificate of Appreciation. Moderators, could you flash the certificate? And may I call on Sir Ray Esguerra, the Chief of the Environment and Biotechnology Division, to do the honor of reading the Certificate of Appreciation. Sir Ray. Okay. I think I will be the one doing this one. So, the Republic of the Philippines Department of Science and Technology. Allow me to read the citation. Republic of the Philippines Department of Science and Technology, Industrial Technology Development Institute, awards the Certificate of Appreciation presented to Dr. Lourdes M. Nadala, Balik-Scientist, Diagnostics for the Real World Limited USA, for sharing her expertise as resource person for the webinar entitled Immunoassays, as part of the establishment of the Virology and Vaccine Institute of the Philippines program of the Department of Science and Technology, held online on February 28, 2022 via Zoom. Given this 20th day of February 2022, signed by the Director of DOST-ITDI, Dr. Annabel V. Briones. So thank you very much, Dr. Nadala, for that wonderful presentation. And before we go on, may I ask the participants to actually open their cameras for our group picture taking. So let's go on to the first pane. Smile. Okay. Okay. To the second slide. Okay. To the third slide. To the fourth slide. To the fifth slide. Sixth slide. Seventh. Eighth. Okay. Okay, so that is for our group photo picture taking. And I would like to thank everyone who joined us today, and especially to our resource speaker for sharing her time and giving her valuable insights into the immunoassays. And for the continuation of the VIP webinar series, we will be keeping you posted for the other or succeeding webinar series or webinar programs that will be showcased by the VIP or the Virology team of the DOST Science and Technology, Department of Science and Technology. So again, thank you all to those who joined our learning session for today. And please answer the evaluation form flashed on the screens to secure a copy of your certificates. And again, this has been Lendley Susie from the DOST-ITDI Virology team. Please do like and follow Environment and Biotechnology Division ITDI Facebook page for announcement regarding future webinars. Happy lunch and enjoy the rest of your day. Thank
you. Bye-bye.
The
Kilaok na ang manok, hudyat na ng pagpasok. Paglilingkod na walang kapalit, sa bayan ng aming hatid. Tara na, kaibigan, huwag kang magpaiwan. Gamitin ang dunong bansa'y susulong. Ating abutin ang pangarap ni Juan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas.
Anumang hamon ay haharapin, mahirap man ay kakayanin. Sa pinagsamang lakas at galing, tagumpay ay mararating. Tara na, kaibigan, huwag kang magpaiwan. Gamitin ang dunong bansa'y susulong. Ating abutin ang pangarap ni Juan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas. Ating abutin ang pangarap ni Juan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas.
Ang pangarap ko ay maging piloto.
Pangarap kong magkaroon ng chief alternative for personal hygiene.
Pangarap kong magkaroon ng mabilis at murang transportasyon para sa lahat.
Pangarap kong masagot ang malnutrition.
Pangarap ko pong magkaroon ng effective communication means for emergency.
Pangarap kong ma-maximize yung renewable energy source and to reduce the carbon dioxide emission.
Pangarap ko pong maging scientist.
Ngayon na o simulan na, humanda sabay-sabay akyat. Hawak kamay tayo'y angat, lipad. Ating abutin ang pangarap ni Juan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas. Ating abutin ang pangarap ni Juan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas. Maayos na bukas para sa Pilipinas. Maayos na bukas ating iparanas sa bayang Pilipinas. Ating abutin ang pangarap ni Juan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan.
Kilaok na ang manok, hudyat na ng pagpasok. Paglilingkod na walang kapalit, sa bayan ang aming hatid. Tara na, kaibigan, huwag kang